Cancer plasticity is this process where when you get continual therapeutic pressure, for example, from a drug, these cancer cells essentially will alter their identity and find new ways to evade that therapy. And with that becomes more aggressive. And in fact, this can be seen in a number of cancers, including lung, gastric, and prostate cancer, which is usually associated with, unfortunately, very poor prognosis and overall survival. We know in a tumor is not just the tumor cell itself, but it's surrounded by a number of other supporting cells called tumor microenvironment. These can be vessels that supply blood to the tumor. They can be this particular type of cell called a fibroblast, which actually lay the matrix that the tumor cell lays on. And then there's the immune cells that survey usually foreign particles and try to get rid of them. So as the cancer cell changes and finds ways to evade these therapies, the tumor microenvironment or the tumor ecosystem actually changes. So our lab studies this topic of therapeutic resistance, and one of the things that we've become interested in is to try to understand the interplay between the tumor microenvironment and this process of cancer cell plasticity. And so one of the questions is, how do you actually target those aggressive states, and how do you create systems to be able to see what programs are unfolding over that cascade? And if we can understand this interplay, can we actually create targets to be able to alleviate that stress and that resistance that emerges. We, in a collaborative study, found that there's particular pathways that are enriched, and those can be blocked. So genetic programs that become activated and you can actually block them. And one of them is around this inflammatory program called JAK-STAT signaling. And in fact, if you take FDA-approved drugs that block that pathway, you can actually reverse this whole process and resensitize those cells to the initial therapeutic drug that you put on at the beginning that they became resistant to. That's one of the foundations of the lab, is to try to take that particular finding and expand it to try to understand the other pathways that may be modulating plasticity. We are using a number of tools, including taking patient samples and generating 3D models of them. These are organoids. We also use more classic forms of genetically engineered models to really look at the dynamic unfolding of this process. And more recently, we've also implemented really next-generation sequencing tools to look at a single-cell resolution each of the particular features within each of the tumor cells. And we can see that there's a lot of heterogeneity. And one of the questions is, how does that heterogeneity actually unfold, and how can you target it? The ultimate goal is to try to translate this from the bench to the bedside. And so we have a really exciting trial at Yale in collaboration with Memorial Sloan Kettering that looks at trying to block an inflammatory pathway to reverse cancer cell plasticity. And so the hope is that if we can uncover this within the laboratory, we can actually bring new targets forward that would help our patients.